Okay, so if the exam is going to be next Friday, it will start from the cell structure and function slide set on the details of peptidoglycan moving forward. So that's chapter 3. And then we got into chapter 13 on the intro ideas of metabolism, glycolysis, and the TCA cycle and the electron transport chain. It'll cover through what we're getting into this today. And then on Monday, I'll make a firm cutoff point of where you can stop for the second exam. And so then I want to go over the stats of the first exam. And then I'm hoping in Monday's lecture time period, I can go over some of the questions that if I see, a bunch of people miss this question, I'd like to go over that with you. But remember, if you want to go over your first exam in detail, make an appointment with me and I'm happy to do so. Okay. I think one-on-one works way better. So you can get a little bit more fine-tuned advice. But on Monday, I will talk to you as a class about the first exam and some general ideas to do better on the next one. Which, of course, one of the huge ideas is to be studying a little bit every day. Really focused bursts, short time periods of really active study is the way to go. The neuroscience has proven it. So, questions on the second exam? Yeah. Question? Anyone? Question? I think we left off here. So let's see. All in all, when you look at what's happening in this figure, what kind of strategy are the microbes using in general? What can you say about what's being drawn in that figure? Like what's going on there? Electron transport. Okay. So electrons are going from donor to acceptor. The way they drew this, who is the terminal or ultimate electron acceptor? You can just say the letters or numbers. N20, the ultimate electron acceptor, down to N2. So N20, is that oxygen? No. So what kind of metabolism is this? Anaerobic. There you go. Anaerobic metabolism. So, this is specifically called denitrification, this whole process that's going on. So Pseudomonas is really good at doing this. So denitrification is the idea of taking nitrate, refining it, and pushing it all the way to dinitrogen. And so that's been heavily studied. Pseudomonas is the only one who can do that. But again, if you are a grower and trying to grow plants, it's not really what you want to have going on in your soil because plants love NO3 minus. And so if you keep depleting that in your soil, your plants are going to be not happy. So you don't want a ton of this going on. Although Pseudomonas, there are plenty of Pseudomonas species, by the way, that are very beneficial towards plants and can be, we call them plant growth promoting microbes. And so there certainly are, but on the other hand, there's some really deadly Pseudomonas that can kill plants. So it just depends what species you're looking at. So this idea of electron transport chain processes, just looking at a few, we have so-called aerobic respiration left, where you see oxygen as the terminal. So aerobic is referring to that idea where you have, where's oxygen sitting on the end there? Then nitrate reduction is being shown. So again, with nitrate reduction on the top right, you are still seeing the general idea of an electron transport chain process. And so you have the NO2 minus, okay. Then denitrification is that big long one on the bottom. But again, you can see there's these themes of electron transport chains, donor to acceptor. They're all embedded in the membrane like that. And it just depends on whether or not you're going to see oxygen at the end or something else, whether you call it aerobic or anaerobic metabolism. Okay, let's think about another idea. Here's another one, chemolithotrophy. Although very often what we do is we get rid of the front part, the chemo word. Chemo is just referring to chemicals. And so that's often like, yeah, that's a given. It's kind of, overkill. And so people will shorten it down to lithotrophy, which I'm always a fan for shortening words. And so here we're looking at the idea of inorganic compounds as energy sources. If you look up litho, it means rock. If you look up trophy, it means to use or to metabolize. And so lithotrophy is eating rocks. That's how I remember that we're talking about inorganic rocks or inorganic compounds. And so this idea that they can eat rocks, so to speak, in order to get their energy needs met. And so they're using those inorganic compounds as electron donors. And we can't do that. We don't have this ability. And so they're using them as electron donors and as an energy source. So we're going to see as we work through all the physiology topics. Basically, when you look at microbes and you're trying to figure out how are they staying alive, you look at what are the electron donors. That's one of the things. Then you look at energy source, and then you also look at carbon source. Those are the three big things you're looking for. What's their carbon source? And then where are they getting their electrons from? Where are they getting their energy source? Yeah, yeah. Some of them can do that. Others were feeding off of the grime, like the carbon, so organic stuff, the grime that had coated the rocks too. The marble sculptures. Yeah, but some of them were capable of doing this, and you have to be careful with those because you can't take it too far, otherwise they start to degrade the marble. So microbes that do this, they can have lots of different resources for obtaining these inorganic compounds. Hydrogen sulfide is organic. That's a favorite one that microbes like to use, and that's coming from a lot of volcanic activity. One eruption of a volcano releases massive amounts of hydrogen sulfide into the atmosphere that can circulate globally. All of our agricultural practices, mining practices, we certainly can make it so that microbes have easier access to nitrogen and iron compounds as well as others. Just our action of burning fossil fuels creates inorganic compounds that they can use, industrial waste. So we humans are like one of the biggest sources in terms of how we interact with our environment and all of our industrial practices, as well as volcanic activity. And then other microbes in the area. So some microbes near these lithotrophic organisms are busy doing their thing. And as they're getting their ATP needs met, they're producing hydrogen sulfide or they're producing hydrogen gas or ammonia. And then they excrete that out because it's just waste to them. They're trying to play the game to get ATP. In the process, they make hydrogen gas and then they just excrete that out. Then a lithotrophic organism goes, hey, I can use that. And they'll take that strip electron from. So in diverse groups of microbes, you can have that flow of nutrients between the microbes where the waste of one is the treasure. What is that saying? The one person's trash is another person's treasure. Very much that's the case in microbial realm. You think about natural settings. that as one microbe excretes out waste, there's other microbes nearby going, I'll take that. That's great. It's something I need. And so all of these are situational depending on the environment. But if you think about what are their carbon needs, how are they doing that? Most of them actually take in CO2 and they use the carbon of carbon dioxide as their carbon source to build their organic molecules. And then some are what we call heterotrophs for carbon. What that means is they have to take in organic molecules, not CO2. They'll take in, so we're heterotrophs. We have to take in organic molecules as our source of carbon. We can't just grab CO2, unfortunately. And so if a lithotrophic organism, microbe, uses an organic carbon source, then they start referring to them as mixotrophs because they'll strip electrons off of an inorganic, but then they're taking carbon organic. It's kind of weird. So they're mixing it up a little bit here. And so they're pretty rare. There's not a ton of them out there that we're aware of, but they do exist. So lithotrophy, they're taking electrons off of inorganic. So here's a table showing the different options, and this is not a complete table. And so you can see electron donors in that second to last column, right? They're looking at who's donating electrons. So you can see like the first 3 rows, it's hydrogen gas, then iron, then ammonia. Okay, so you can see a range of different sulfur compounds as well. So the microbes are capable of stripping electrons or taking electrons off of those inorganics. So they're oxidizing those inorganic molecules. And then you can see a little bit, they're hinting at, well, who gets those electrons? Because remember, it's a redox reaction. Something's going to donate, something has to accept. And so here you're seeing that there's a range of electron acceptors as well. Okay. Let's look at this. Okay, so we got several things going on here. In general, when you look at this process, again, what could you say in general, what's going on here? Yep, and how is that proton gradient getting, like what's happening to create proton gradient? Electron transport again, okay? So that's all accurate, what you all just said. All right, let's try a iClicker. Let's see if we can work through. See if I can minimize that thing. That. Yeah. Okay, so what is donating electrons into this electron transport chain? So now you have to look for, let me see if I can get this out of the way. Are you still able to get in? Okay, that was weird. I don't know. It seems to be working. 10 more seconds. Are we all clicked in? Okay. So what is donating electrons? Wow, 100%. When's the last time that happened? I made it too easy. C, that is correct. So we have this idea of the hydrogen gas coming in, top left. It's not gonna let me draw. And it's like the kickoff of the electron transport chain. So that transmembrane complex here is getting those electrons, stripping those electrons of hydrogen. It goes through that electron transport chain process. Okay. Let's see if I can. Oh, by the way. So why is A and D, which you didn't get to see, I forgot to click on to show D. Why is A and D not an option? Yeah, they don't got any electrons to donate. They're like fresh out. If they're in the plus state like that, it's not gonna happen. Okay, let's see if I can advance. Okay, so I think those are your only two options for this question. Ten more seconds. So what is the nature of hydrogen gas? It is organic. Sorry, inorganic. Click on it and say the wrong word. It's inorganic. Hydrogen gas is inorganic. Okay, so keep that in mind. It's inorganic. Ah. 10 more seconds. So 78% said oxidative. That is correct. So why is that correct? Oxidative phosphorylation. It's stripping electrons, right? So oxidative doesn't always mean oxygen. In this case, there is oxygen involved, but remember, it doesn't always mean that. But oxidative means stripping of electrons so that you have this redox reaction in order to generate ultimately ATP. So oxidative phosphorylation is really, you should think of it as electron-linked phosphorylation, electron chain process linked, electron transport chain process linked phosphorylation. Why is it not substrate level phosphorylation? Do you see a phosphorylated substrate anywhere? No. Okay, so glycolysis, there's a couple of places there where there's a substrate in the glycolysis pathway that has a phosphate that gives it off to ATP. Here you don't see it. You see there's an inorganic phosphate Forgot to put that in there, I guess. But there's an inorganic phosphate just floating around there that will attach to ADP once you have the protons flowing through the ATP. Okay, so those theories of, oh, there was, I don't know, there isn't, so. Okay, so overall, this process is called whoops. So pick the best answer. Ten more seconds. Okay, 70% said lithotrophy. And that is correct. That's the most precise answer. So lithotrophy is the idea that you have inorganic compounds as your electron source. And that is certainly fitting what we see here. Let me draw. We already said H2 is inorganic. And so we have an inorganic that's getting stripped of electrons. That's lithotrophy. And yes, there's oxygen involved here. But the more precise way to think about this is you have to look at who's the ultimate electron donor too. It's not glucose. It's an inorganic. It's hydrogen gas. So these series of iClickers are the thought process that I use when I'm trying to figure out what I'm actually doing physiologically. Like if I'm presented with something like, what's going on here? I look for, well, do I see an electron transport chain? Or do I see substrates in a pathway like glycolysis? Who's the ultimate electron donor? Is there a terminal electron acceptor somewhere? Is there oxygen or not? Who's sitting up, it could be oxygen at the end, which it's the best electron acceptor. But as we know, microbes can put something else there. So if a microbe put nitrate there instead of oxygen, then we say, oh yeah, but really lithotropy. We're looking at this idea of inorganic electron donors. So let me go back to where did it go? Oh, here. So this table, hydrogen gas is a favorite. Microbes usually have hydrogen gas around them due to a variety of things happening in their environment, including other microbes. So there could be other microbes nearby that end up making hydrogen gas and excreting it out. Do you remember what kind of metabolic process is kind of hydrogen gas is a favorite product to make in order to play the game? Do you remember? Fermentation. Yep, fermentation. And so fermentation, there's going to be a lot of microbes in consortiums that are doing fermentation, and one of the favorite things that they like to do is give electrons off to protons, because they usually have a lot of protons around somewhere, and they can just give the electrons to a proton and end up making hydrogen gas, but they don't care about the hydrogen gas. It's the game they're playing for redox balance. So they get rid of that, and then a lithotrophic organism nearby say, I'll take that. That's my source of electrons. And so hydrogen gas is also pretty high up there on the redox tower. Very capable of giving off and donating off electrons to all kinds of other things. And so it's a smart move for microbes. If they can do lithotrophy, then look for hydrogen gas. That's a really favorite situation. But you can see here that, well, and then this is technically hydrogenotropy, which is a lithotropic reaction because hydrogen is being stripped of electrons. So this table of describing different ways to do hydrogenotropy, say that word, and then this is one example. Okay. So lithotropy, they're extracting or stripping electrons off of something inorganic. There's some other kinds, like taking some different nitrogen compounds, like ammonia or nitrite. So that can be abundant in certain environments for microbes. And so those nitrogen-containing compounds are often used in lithotropic reactions. So for example, there's these groups of microbes that are called nitrosifiers, and I wrote down a couple genera. You don't need to memorize that, but knowing nitrosifier term is a good idea. And so the nitrosifiers, what they do is they take ammonia, for example, and they strip electrons out. Well, ammonia is inorganic. We're doing lithotropy. So as they do that, they'll end up making nitrite, NO2 minus, nitrite, which they'll then get rid of. And then another nearby group of microbes, which they haven't given a group name to, which I don't know why, but they don't. So something like nitrobacter could be nearby. And they'll say, okay, you excreted out that nitrite. I'll use that though, and I'll do my version of lithotropy. And so they're going to strip electrons off nitrite, and then you get nitrate, NO3 minus, NO3 minus, nitrate. And so this process from the first bullet point to the end there, that is called nitrification. Okay, so taking ammonia in this case, nitrite to nitrate, that process is called nitrification. So very often what you'll see is some microbes in a mixture in a real environment, like in soil, they'll do nitrification. And then other microbes right nearby might do denitrification. And so if you put it together, it might look something, this is how you think about those two. So nitrification versus denitrification. Here they're showing the nitrification with the red arrows that we just looked at, okay, taking it to nitrate. But then other microbes nearby will say, oh, you just excreted out nitrate. That's good because I'm going to use that, and they use that as a starting, and then they take it in this direction. So denitrification is taking nitrates and following the blue line, the dinitrogen. And so having this in these environments, like a soil environment where this stuff is going on, you can see how now, groups of microbes that are doing physiology can really benefit from hanging out together, because the waste of one microbe is going to be the treasure, the food source, electron source of another nearby. And so, and then, these can get out of whack. Like I said, I really wouldn't want denitrifiers in the soil trying to grow plants. But on the other hand, sometimes it can be, we have to have recycling of nitrogen. We got to get enough nitrogen back into our atmosphere. Otherwise, everything's going to go wonky. All right. So microbes are the ultimate recyclers. And, without them doing their thing, the Earth wouldn't be the way it is. We wouldn't be here because they're just so good at recycling the nutrients. Okay. So here's another example of lithotrophy. It's called ANAMOX for short. It stands for anaerobic ammonia oxidation. So the AN is anaerobic, AMM, ammonia, and OX, oxidation. anoxic, anaerobic, same thing. This is the reaction. So they take ammonia. That should actually be, yeah, it's ammonium. Ammonium and ammonia are interchangeable, reversible reaction. Reacting it with nitrite, NO2 minus is nitrite, and then you get that dinitrogen and water. So here's another process of this one's pretty unique. There's so far anyways, they've only discovered a handful of microbes that can do this. But if you can do this, they've calculated the free energy that you get from running this reaction, and it's negative 307. What do you need for ATP? Negative what? Negative 31. Let's go with what the book says. Negative 31. So that's pretty good. You get a good dose of free energy here. So these types of microbes, when they run this reaction, and it's just an electron transport chain reaction that they're ultimately running, but it is giving them a lot of free energy to make ATP. They don't care about the dinitrogen. That's a gas. It just gets diffused out of them and it goes back into the atmosphere. So they're helping to recycle that. So if you think about ammonia and where humans make use of this, like wastewater treatment plants, when you flush the toilet and it goes to the wastewater treatment plant, so a lot of that is urine, and urine has ammonia. And so what you have here is that process of in wastewater treatment plants, they deliberately create anaerobic environments to get this reaction cooking so that you can clean up the water. And so this can actually be very, very useful in wastewater treatment plants for cities, to just use the microbes. And microbes do it as long as you cultivate and give them the right, anaerobic environment and get them going. They can feed off of our waste very nicely, clean up our water, and it's cheap. You just have to keep those microbes happy. That's all. They're also found in anaerobic environments in the ocean and are critical players in the ocean for, again, recycling nitrogen and getting that dinitrogen back up into the atmosphere. So they're kind of funky organisms because when you look at them under the microscope, we keep saying that bacteria don't have internal organelles. Well, it's with more and more study, that's not exactly accurate in the sense that, for example, this anomopsisome idea, it's really, when you look into these bacteria inside, they have all this membranous material inside their cell. And so it's like, well, technically that's like an organelle. And so it's getting harder and harder to say that prokaryotes don't have organelles. And this isn't the only example. I'll mention a couple others. So they have a very unusual physical nature in the sense that they're just chock full of these membranes. And that's where they put all the enzymes necessary to run this reaction. So they're really devoted to running this pathway at very high levels, high intensity, with the way they're physically structured. Here's another funky one. You know what genesis means in general? Creation, right? So genesis means to create. So creating methane is how you would interpret that word. And so here we have this idea that some microbes can take, for example, CO2. They can take other things, but CO2, they can use a few other molecules, either single carbon or two carbon compounds as their starting point, and they'll end up making methane off of it. And so as far as we know, it's only cells or species in the domain Archaea that can do this. And they give them a special name. They say they're methanogens because they're generating methane. So here's the simplest way that they do it. They'll take CO2 and react it with hydrogen gas. And as they do that, they'll create methane, which they don't care about. So the methane just is a gas, it gets excreted. So what they're doing is they're trying to make ATP. And so they get enough free energy from this in order to make ATP. But they just excrete out the methane. Then nearby, hopefully, there's other microbes that aren't methanogens. They're trophs. So they're called methanotrophs, which means they're going to use or eat the methane. Okay, so methanotroph means that they're actually eating or using methane. And so they can be side by side. As soon as the methanogens go through their reaction, excrete out the methane, methanotrophs say, I'll take that and I'll use it as a source of electrons. And so they're running like an electron transport chain. TEA is a terminal electron acceptor. And so what that, anytime you hear that, terminal electron acceptor, we're thinking about an electron transport chain process. And depending on the microbe, they can put oxygen at the end, which would be the best, but they can also do others. If they're in an anoxic environment, they can pick something else. And a lot of them are in anoxic environments, but not all. So methanogens, these archaea, which we may have some of them in us right now. There are a handful of archaea in our gastrointestinal tract, and they're found in other animals' gastrointestinal tract. But you can find them a lot swampy areas, marshy areas, lake sediments, paddy fields. Find them. Cows and cattle have probably been the best studied in terms of the methanogens that live in them. So they live in the rumen, which is part of the gastrointestinal tract. But they can be found in the cecum of horses and rabbits. So one of the longer term studies that I'm aware of is when they actually operate on cows. And they actually keep the cow alive, but they essentially put a hole in the side of the cow and they cap it off. And then they can go in and uncap it and go in and dig some of the contents of the rumen out and study it. And so these are long-term studies. They keep the cow very happy and healthy. And otherwise, it's just being used for sampling the microbial ecology of the rumen. Have you seen this before? So the one that I'm aware of is down at Michigan State University. I know that they used to do this. I think they still do. Got a picture of Dan. He graduated from our master's program, but he was down at MSU. He took my microbial ecology class, and we were talking about this. He's like, "Yeah, I actually got to do this." And you're allowed to. They take certain people that are allowed to go in behind the scenes, and he actually got to go in and sample. I don't know if I'd want to do that. He doesn't look too happy about it, but he said it was cool, good experience. So these methanogens and methanotrophs. So here you're seeing methanogenic reactions. So CO2 is probably most straightforward, but they can use methyl compounds. See, there's a range of select compounds. But no matter what one they're using, whether it's carbon dioxide or something else, basically what they're doing is they're running an electron transport chain process. And that's going to get them their proton gradient. Remember, it's linked. Electron transport chain, proton gradient. And so they've not discovered any, even though they're in anoxic environments, they don't see any doing substrate level phosphorylation. They just don't seem to do that. So it's always this electron transport chain process idea. Okay. Okay, so that is a sampling. I did want to also get into how some microbes use light. And there's a couple of different ways. Here, this is the use of light in order to make ATP, but technically you can't call it photosynthesis. But these microbes are interesting because they have a special protein. So the most well-studied is Halobacterium salinarium. So when you see salin, what do you think of when SALIN? What does that make you think of? Saline, salt. So they named it that because Halobacterium lives in super salty environments. I actually like growing this in the lab because you don't have to be sterile at all. Because you make the agar plates so salty, nothing else can live. So it's like you don't even have to, you just have to heat up the agar well enough to get it to melt and pour, and you don't have to worry about it. The other cool thing about this, working with this, when I used to teach BI 100 to non-majors, biology to non-majors, I wanted to get across the point of DNA. And so I wanted them to visualize, how much DNA is in microbes. So I took this and gave them test tubes, and all we had to do is put in pure water, and it made the halobacterium essentially explode because they're so salty, and then you expose them to pure water, it's sort of osmosis, and so they'd explode and release all their DNA raw, and you just see it precipitate just with the naked eye. So it's a fun microbe to play around with, but they're also really cool because they have this bacteriorhodopsin protein that they stick in their membrane, and then they have a retinol that is bound to it. Okay, so this should sound vaguely familiar because our eyes have some similarities here. And so the retinol is a pigment, carotenoid-like pigment, and it absorbs light energy. And so what's going on here is that you get a very small reaction going on here, but it is tied to the idea of a proton gradient, or the fancy way is proton force. So essentially, you see proton gradients developing when light is absorbed by the retinal pigment. And if they do that enough, they're going to get ATP through that proton gradient. So these microbes can hang out in really, really salty, like if you've ever flown over California and you see those different colored salt ponds, essentially, where they can be bright orange, pink, a lot of that's halobacterium in those salt ponds. They love that. And so here you're seeing the idea of what we just talked about visually. So here's the retinal, and what they're showing here is when the retinal is getting hit with light. So here, HV 570, that's 570 nanometers of sunlight energy, which is what? Green, I think, greenish colors. Anyways, the 570 nanometers of sunlight energy strikes the retinal, and that converts the retinal from the trans form of the pigment to the cis form. Is anyone else shuddering with organic chemistry references here? Trans and cis. Ooh. So that shift, that physical shift, allows for proton pumping. And so you keep hitting that bacteriorhodopsin with the retinal, and you get that shift, and you get a proton pump. And over and over and over, and until you get a lot of protons being pumped out, and you get that gradient. And then the protons diffuse back through the ATP synthase. So really quite a simple little mechanism in order to create a proton gradient, which is quite unusual. And so this is a pretty ancient mechanism. It's one of the first ones that evolved in order to use light energy to accomplish getting some ATP. Okay, we got to stop there. So the next topic we're getting into is how we see some microbes doing their version of photosynthesis. So we'll start that on Monday.